Welcome to your personalized deep dive. Today, we are plunging right into the computational heart of modern biology, that's bioinformatics.
Yes, and our mission really is to unpack how researchers make sense of just oceans of biological data.
We're working from some foundational concepts and protocols, mostly from around 2008 or so, but they show the analytical engines that really power discovery.
Absolutely, it's a fantastic snapshot. We're talking methods that stretch from, modeling a single RNA strand folding.
All the way to drug design and disease diagnostics. It's quite the range.
It is, and these computational approaches, they underpin fields like toxicogenomics.
Prion protein research, too, I saw. Even methods developed for SARS and other coronaviruses, tissue engineering.
Environmental genomics, it's everywhere.
It really is the infrastructure.
Right. And the core challenge, fundamentally, is dealing with data that's massive, diverse sequences, structures, expression levels, and pulling out actual knowledge.
So our sources kind of split this. Volume ion data, sequence evolution.
And volume 2, hitting structure, function, and those applications. So let's jump in. Where do we start? Predicting the physical world.
Okay, let's tackle RNA folding first. That seems like a beast. We know the sequence A, U, C, G, but getting that 3D shape. The sources say just finding the minimum free energy, the MFE. Well, it's often not enough. Why is that?
Well, the issue is thermodynamics, really. Equilibrium. You can have many, many different folds with energies that are incredibly close to that MFE.
So the best single fold might not be the whole story biologically.
Exactly. If you only calculate that one optimal fold, you might miss the reality, which is often a whole collection of structures kind of flickering in and out of existence, a dynamic balance.
So you can't just hunt for the single winner.
Yeah.
This is where software like UNAFold comes in.
That's the idea. UNAFold computes not just the optimal, but also suboptimal structures, a collection of them.
How does it do that?
It uses partition functions. Think of it like instead of finding the best structure, it calculates the statistical probability of every possible structure. And that lets you generate these probability dot plots, or PDPs. You're not just getting a structure, you're getting a map showing how likely specific base pairs are to form. It's about confidence.
Right. Now, the sources had a really specific warning here. For folks using microarrays.
Yes, that's crucial. You can't just assume the standard rules for molecules interacting in solution apply perfectly on a microarray.
Why not?
Because one of your molecules, the probe, is stuck down. It's immobilized on a surface.
Okay.
So calculating things like solution concentration gets really tricky. You have to think about diffusion, how long it takes things to find each other. Kinetic issues become much more important. It's a heads up for anyone doing that kind of experiment.
Good practical point.
Okay.
Moving up in scale, from RNA to proteins. If we need a 3D protein shape, but we can't get it experimentally, the go-to method seems to be comparative modeling, CM.
Yes, CM, often called the elective method. And the logic is pretty straightforward. Proteins with very similar amino acid sequences, they almost always fold up into similar 3D shapes.
Makes sense.
So if you can find a protein with a known structure, that's your template, that lines up well with your unknown protein sequence, your target, you can build a pretty good model.
And finding those templates, that's where tools like BLAST come in. Sequence comparison.
Exactly. You use BLAST, or similar tools, to search vast databases of known sequences and structures.
And if you're using BLAST, the key number everyone talks about is the E-value. The expectation value. What exactly is that telling us?
The E-value is critical. It tells you how many alignments with a score equal to or better than the one you found would be expected to occur just by pure chance in a database of that size.
So a lower E-value means it's less likely to be random, more likely a real match.
Precisely. But there's a trade-off. If you set your E-value threshold really, really low, say, demanding near certainty it's not random,
then you reduce your false positives, fewer incorrect hits.
You do fewer incorrect hits, but
you might throw out real, meaningful connections, ones that are maybe more distantly related evolutionarily.
that's the risk. You increase your false negatives, you miss true homologs because their sequence similarity has just drifted below your strict cutoff over millions of years.
So it's a balancing act. Finding that sweet spot for the E-value.
It really is. It's one of the fundamental skills in bioinformatics, managing that trade-off.
Okay, and because no single prediction method is perfect, the field moved towards using meta servers, like getting a second, third, fourth opinion.
Exactly. Wisdom of the crowd applied to structure prediction. Systems like 3D Jury or MetaBasic, they don't do the prediction themselves.
Why do they do that?
They act like managers. They send your sequence out to multiple different prediction servers, collect the results, evaluate them, and then combine them, often based on consensus.
Now that worked better.
Generally, yes. These meta-servers often outperform any single method they draw upon. They give you a more robust, higher confidence prediction.
All right, so let's say we have a decent model of the structure. The next big question is, what does this protein actually do? Its function.
Right. And the principle here, according to the sources, isn't just finding one link. It's about guilt by multiple associations.
Meaning you need several different lines of computational evidence all pointing the same way.
It's exactly. Convergence is key. You need independent analyses all suggesting the same function.
And one really neat technique for finding functional links mentioned is the Rosetta Stone method, looking at fusion proteins.
Yeah, that one's fascinating. It leverages evolution's occasional efficiency moves.
How so?
Well, sometimes you find two proteins that are separate entities in one organism, maybe like in humans.
Okay.
But in another organism, say a parasite or bacterium, those two proteins are actually fused together into one single continuous protein sequence.
Like they've been stitched together.
Precisely. The classic example is DHFRTS in the malaria parasite P. falciparum, dihydrofolate reductase, and thymidylate synthase. Separate in S, fused in the parasite.
So what does that fusion tell you?
It strongly suggests those two originally separate functions are tightly linked. Maybe they physically interact, or they're consecutive steps in the same critical pathway. Evolution wouldn't likely fuse them unless there was a strong functional reason.
That's clever. Using evolutionary quirks to deduce function. What about the gene neighbor method? That sounds more about location.
It is. It looks at gene order in prokaryotes, mostly. The idea is simple. If you look across different evolutionarily distant bacteria, genes that consistently stay physically close to each other on the chromosome are very likely to be functionally related. They might be part of the same operon or pathway.
Does it matter which DNA strand they're on?
The signal is strongest when the neighboring genes are on the same strand, suggesting they might be co-transcribed. But even conserved adjacency of related genes paralogs can be informative.
Okay, so we have fusion proteins, gene neighbors, and the third big one is phylogenetic profiling, co-evolution.
Right. This method looks for patterns of presence and absence across many different species.
Explain that a bit more.
Okay, you take your protein of interest, protein A, you then check across, say, 100 different reference genomes. Does each species have a homologue of protein A, yes or no? You build a profile, like a barcode, of its presence-absence pattern.
Got it, like a fingerprint across species.
Exactly. Then you do the same for protein B. If protein B has a very similar phylogenetic profile, it tends to be present in the same species as protein A and absent from the same species. That suggests they are co-evolving.
And why would they co-evolve?
Because they likely depend on each other. They might be part of the same molecular machine or steps in the same pathway. If you lose one, the other might become useless, so they tend to be gained or lost together during evolution.
And comparing these profiles, the sources suggest using more advanced math, not just simple correlation.
Yeah, they recommend things like mutual information. Simple correlations might miss more complex nonlinear relationships. Mutual information can capture those better, giving a clearer picture of the functional linkage.
That's a powerful set of tools for inferring function. Let's shift gears now to applications and a big challenge that came with microarray data, computational diagnostics, and this issue of overfitting.
Ah, yes, the curse of dimensionality. This hit hard with gene expression data.
What is the core problem there?
It's a fundamental statistical mismatch. You're measuring thousands, maybe tens of thousands of genes. Those are your features.
Right.
But you often only have data from a relatively small number of patients, maybe 50, maybe 100. Those are your samples.
The features vastly outnumber samples. Why is that bad?
Because with so many features, it becomes statistically easy to find a combination of genes that perfectly separates, say, your cancer patients from your healthy controls in your specific data set.
Okay. Sounds good so far. Perfect separation.
It sounds good, but it's often fooling yourself. The complex signature you found might just be fitting the random noise and specific quirks of your small group of patients.
Ah, so it doesn't generalize.
Yes.
It fails on new patients.
Exactly. It overfits the training data. You get 0 errors on your initial set, but it completely falls apart when applied to new clinical data because it learned the noise, not the true underlying biology of the disease.
So how do you combat that overfitting?
You need methods to force simplicity. Things like regularization, which penalizes overly complex models, and robust validation techniques like k-fold cross-validation, where you repeatedly hold out parts of your data to test the model's predictive power on unseen samples.
And beyond just avoiding overfitting, researchers want the best, simplest signature, right? The sources mention things like the min feature set problem.
Right, that gets into combinatorial optimization. Can we find the absolute smallest set of genes that still does a good job discriminating between patient groups and is robust across different subsets of the data?
Why smallest?
Smaller, simpler signatures are generally more likely to be biologically meaningful and, crucially, more likely to work reliably when validated independently, say in a different hospital or population. They're less likely to be built on noise.
Okay, another huge application area, drug discovery. Specifically, virtual screening using molecular fingerprints.
Yes, molecular fingerprints became workhorses. They're essentially bit string sequences of zeros and ones that encode the presence or absence of specific structural features or fragments within a molecule.
So you turn a molecule structure into a barcode.
Pretty much. And then you can rapidly compare the fingerprints of millions of compounds against the fingerprint of a known active drug. Similar fingerprints often mean similar structures, which often means similar biological activity. It's a way to computationally sift through huge libraries.
And I saw a specific algorithm mentioned, MAD, determination and mapping of activity-specific descriptor value ranges. That sounded a bit different.
MAD is interesting because it tries to find remote similarities. It's not just about overall shape overlap like simple fingerprints.
How does it work?
It analyzes a set of known active compounds and tries to identify which specific ranges of chemical property values, descriptors like size, charge, flexibility, are actually critical for the observed activity.
Ah, so it pinpoints the activity-determining features.
Exactly. Then it screens new compounds, looking for ones that fall into those same critical descriptor ranges, even if the overall structure looks quite different. It helps find molecules that traditional similarity searching might miss, boosting virtual screening success.
Very cool. Okay, final challenge. Just dealing with the sheer volume of information out there, especially in published papers. Text mining comes into play.
Absolutely essential. The biological literature is enormous and growing exponentially. We need computational tools to help us extract knowledge from it.
What kind of tools?
Well, some early systems like PubGene took a simple approach. If two gene names appear together in the same Medline abstract, infer a potential relationship. Rank them by how often they co-occur.
A bit crude, maybe, but a starting point.
A starting point, yes. But accuracy relies heavily on something called named entity recognition, or NER.
NER, what's that?
That's the crucial step of correctly identifying mentions of genes, proteins, drugs, diseases in the text, and ideally linking them to specific database IDs.
So it knows which P53 the paper's talking about.
Precisely, it disambiguates. Is it the human P53 gene? The mouse protein? Linking text mentions to concrete database entries is vital for building reliable knowledge networks from literature.
Okay, so we have all these amazing tools: sequence search, structure modeling, function prediction, text mining. But they're often separate pieces. How do you actually string them together for a real analysis?
That's where computational pipelines or workflows became critical. Think of tools like Taverna or Triana. These were early graphical interfaces for this.
Graphical interfaces. So a biologist didn't need to be a hardcore programmer.
That was the goal. You could visually drag and drop components representing different tasks. Maybe a web service for fetching a sequence, connected to a BLAST search node, connected to a node that queries SwissProt for functional info.
And link them together to automate the whole process.
Exactly. You build the workflow graphically, defining the sequence, choices, or parallel execution of these tasks. It manages the data flow between steps, automating complex analyses that would otherwise require a lot of custom scripting. Huge time saver.
This has been a really fascinating deep dive. We've gone from the nitty-gritty of RNA folding probabilities through piecing together function using evolution's clues like those Rosetta Stone proteins.
Tackling the big data problems in diagnostics, that whole overfitting issue.
Right, and even how we computationally screen for drugs and try to mine the literature. It seems like the core skill running through all of this is confidently pulling together insights from really different kinds of data.
Absolutely. Synthesis is key. And we spent most of this dive talking about methods to find those functional links or associations. But how confident can you be? Often, the highest confidence comes when completely independent data sets converge on the same answer.
Like combining protein interaction data with gene expression data.
Exactly. And the sources hint at something interesting. The best results from integrating diverse datasets often come not from assuming they're totally independent nor totally dependent, but somewhere in between an intermediate level of dependence.
Okay.
So here's the final thought for you, the listener. Given that integrating diverse, independent data usually yields the biggest breakthroughs, and highly correlated data sources tend to offer diminishing returns, where should researchers be focusing their efforts now? Where are the truly novel, uncorrelated pieces of biological evidence hiding, waiting to be discovered and integrated to push our understanding to the next level?